I'm Christine Kinnan. As Ros said, I'm the Deputy Director for Education here at ICH, and it's lovely to see you all this afternoon, so many of you, at one of our celebratory inaugural lectures. Okay, so I'm going to introduce Chris to you, and then I'm going to invite Professor David Gadian to give a vote of thanks. As Ros said, this is a very special day in our calendar where we really want to celebrate the works of one of our distinguished professors. So a little bit about Chris. Chris has spent much of his career at UCL. I feel he's really a bit of a homegrown product. Chris started off as an undergraduate in physics. He graduated with first-class honours in 1990. Following on from that, he spent a brief time as a trainee accountant. Maybe he'll tell us more about that. We'll see. He then went on to do an MSc in radiation physics here at UCL. And then he went on to do a PhD in magnetic resonance imaging at our neighbouring institute, the Institute of Neurology, which is just around the corner, which is also part of UCL. So once he'd obtained his PhD in 1998, he went on to do postdocs in Paris and Stanford. I'm sure these are very prestigious institutions, and I'm sure he was very successful. He then got his first academic position at St George's Hospital, which is part of the University of London, in 2001, where he was able to start establishing his own group. Following that, he returned to UCL in 2006 as a senior lecturer, and really he's risen very rapidly, becoming a reader in 2010 and now a professor in 2014. He was appointed as head of the unit in 2009, and with our successful academic reorganisation that we had in 2014, he became section head of developmental imaging and biophysics, which is part of the developmental neurosciences programme. So really, just a little bit about the work of the section. This spans methodological developments in diffusion, MRI, acquisition and image processing, and has many applications in a diverse range of neurological psychiatric disorders of childhood, including brain tumours, sickle cell disease, autism, dyslexia and visual impairment. Now you can see I'm reading this because I don't really understand much about MRI, but I'm sure Chris is going to explain all this to us, and he'll tell us about his work. He's been very successful in raising a huge amount of money through grants, and he has a very impressive list of publications, which again I'm sure we'll hear about. And one thing that I've noted about Chris is he has a very impressive record of PhD students. 9 of his PhD students have successfully completed, which is excellent. But he's also currently supervising 7 PhD students as primary supervisor, plus another 7 as second supervisor. That means he has 14 PhD students. Now, the institute average is 2. Right? So this is an amazing record, and I really think that Chris is doing a huge amount for developing our future generation of biophysicists, and I think that's extremely well done. So I'd like to invite Chris to come up now and deliver his lecture as the new Professor of Imaging and Biophysics, and the title of his talk is going to be Random Walks Through the Pathways of the Mind. Thank you.
So thank you, Christine, for that very kind introduction. In the next 45 minutes or so, I want to take you on a random walk through the pathways of the mind. As we're about to see, magnetic resonance imaging can produce images of the structure of the living human brain that are simply amazing. They've revolutionized our understanding of how the brain is organized, both in health and in disease. I'm going to tell you about the small part that I played in the development of these techniques and what it's like to work in science. Now, just like Match of the Day, this is going to be a highlight show because I've got to compress 20 years of my work in the field into about 45 minutes. So let me start at the beginning. This is my school photo. You can probably recognise me there. A few friends in the audience probably can recognise themselves as well. Now, what teachers say is important. I wasn't doing particularly well at school, particularly in my physics classes. I was getting D for my physics homework. And then one day, my physics teacher, Mr. Trimmingham, wrote these words in my exercise book. I've never forgotten them. You know you're better than this, Clark. It really made me stop and think. And I thought, well, if he thinks I'm so good, then I'm going to put that to the test, which I did. I made much more effort and my grades started to improve. And I think Mr. Trimmingham's words had the desired effect. Wherever he is now, I would like to say from the bottom of my heart, a big, big thank you, because my academic trajectory changed forever after that. And so I progressed through my dippy teenage years to my new romantic phase, somewhat dodgy A-levels, to my arrival at UCL in 1987 to read physics and astronomy. So here I am, the young fresher. After the first year, I actually gave up the astronomy part and focused on just physics after having spent many cloudy and starless nights at the UCL Observatory in Mill Hill. It was a bit like the eclipse this morning for a lot of us. Very, very, what a letdown that was for those of us in the southeast. So here are some thoughts as to why I like physics. Now, it is true that you do have to do some reading, but you don't have to actually remember very much, which is really helpful for someone like me. This was brought home to me by this gentleman, Professor Franz Heyman, who was the head of the physics department and my first year tutor. Professor Heyman was South African. He was an archetypal, absent-minded professor smoking a pipe, usually wearing a tank top inside out. And in one of his tutor groups, I remember distinctly working through a proof on the board. And he was encouraging me with each line of proof until eventually I got to the solution at the end. And I was so thrilled that I'd managed to solve this problem that I said, Professor Heyman, I've got to write this down. I've got to write it down. And he said, in his South African accent, no, you don't need to write this down because you have understood each logical step of the proof. If you understand the logic in each step, you will always come to the correct answer. And they were words that I've never forgotten. So following my physics degree, there was a little bit of an interlude. The country was in recession, sound familiar? And there were very few graduate jobs. Now, I studied physics and the natural world for 3 years, and I was interested in finding out more about the world of business and commerce. And so after many failed applications to a lot of banks, I eventually got a job as a trainee at a firm of chartered accountants. Now there I did learn a lot of very useful skills about management, managing budgets and accounts and so on, skills that have come in very handy ever since. But I do remember sitting in the big open plan office we had and thinking to myself, well, do I really want to do this until I'm 60? And I thought, no, I don't. I want to get back into science. So after a short period teaching GCSE retakes in a sixth form college, I returned to UCL in 1993. And it was the best decision I ever made. I took a master's degree in medical physics, and it was here that I met my wife, Catherine, of course, and it was a great bunch of students. We had a lot of fun. Here I am, all dressed up, ready to go out on the summer ball, and that's actually another chick that was on the course. I started my PhD in 1994 at the Institute of Neurology in Queen Square in this delightful 1970s block that you can see here next to the National Hospital for Neurology and Neurosurgery. And I was up there on the 6th floor. Some are up here, and it wasn't a whole lot better, the building on the inside. Apologies to those of you who work at Queen Square House. I was supervised by these two gentlemen, and I'm delighted they're here this afternoon, Professors Gareth Barker and Paul Tofts, and they taught me all about magnetic resonance imaging. So here is an MRI scanner. Actually, it's our new 3T scanner that's just been installed at Great Ormond Street Hospital for Children next door. Now, I haven't got time to go into how MRI works, but essentially what you need to realise here is that the signal that we get from MRI comes from protons in hydrogen atoms in water. Now, happily, there's lots of water in the body and the brain, so we're able to take really nice pictures. And here you can see a sagittal view of the brain. Now, my PhD was actually focused on this phenomenon, which is called water diffusion. So if you take some ink in a dropper and you release the ink into a glass jar, you'll see that the ink will spread out over time. The ink will diffuse. So here we've got a bunch of water molecules executing what we call a random walk. So just like a drunk leaving the pub, you can see that they're wandering around somewhat aimlessly. But if you consider what physicists call a large ensemble, a large number of water molecules, you'll find that they travel further over time. They'll travel further from their starting position as time increases. And this simple behaviour is described by the so-called Einstein equation, which says that the mean squared displacement here is equal to 6 times the diffusion coefficient times the time t over which we observe the diffusion process. So the parameter I want you to focus on here is d, the diffusion coefficient. As we'll see here, it's simply the mean displacement per unit time. Okay, so this is how we measure diffusion in brain using MRI. So first of all, we've got an image here without any diffusion sensitivity. So the degree of diffusion sensitivity that we apply is called the B-value. Here it's 0. So this is a what we would call a T2 weighted MRI, an axial view. And here we've got bright signal in the CSF. Now, when we make those images sensitive to the effects of diffusion, you can see that there is signal loss, particularly here in the ventricles, where that bright signal now has been rapidly attenuated. Now, in addition to that, to a lesser extent, you can also see signal attenuation in the surrounding brain structure. Okay, so all makes sense, surely, don't worry. So that signal decay behaves as an exponential decay. Okay, so SB is the signal with the diffusion sensitivity, the attenuated image. S0 is the signal without the diffusion sensitivity. B, remember, is the amount of diffusion sensitivity applied, and D is the parameter we want to measure, the diffusion coefficient. So we can simplify that equation by taking logs of both sides, so that becomes the equation of a straight line. So if we plot the log of this signal against the B value, we get a straight line whose slope is the diffusion coefficient, and that's exactly how we measure diffusion. So here we are, here's our graph. The first data point here, no diffusion sensitivity, then diffusion sensitization in attenuated image, and we get a straight line, and that gives us our diffusion coefficient. So that's how we calculate the diffusion coefficient using MRI. And we do that in each pixel of the brain image. So MRI data is just like the images you get from your digital camera. We've got pixels here, and we do that calculation in each individual pixel of this image to build up a grayscale map that represents the magnitude of diffusion in each pixel of the brain image. So you can see here, all the bright areas have got high diffusion. All the darker areas have got a much lower level of diffusion. Now, why is diffusion lower in brain tissue than in the cerebrospinal fluid, this is essentially just free water? It's because of the presence of cell membranes and structures in the brain tissue itself. So this all consists of axons and myelin and so on, which acts as a barrier to the diffusion process, so that the water molecules diffuse along, and then they are hindered and interact with those cell membranes, so that the mean squared displacement per unit time, the diffusion coefficient, decreases. So if you've all followed that, I hope you have, you can see that this method provides us with a quantitative parameter that reflects the overall amount of tissue structure present. Now, another interesting phenomenon is when we look in the white matter of the brain. So in the white matter, we've got long, thin axonal structures which connect the different cortical regions together. So here are some long, thin axonal structures, and water can diffuse more easily along these pathways than in directions perpendicular to these pathways, because along the pathways there are very few barriers to the diffusion process, whereas in this direction perpendicular to these white matter tracks, there are a lot of barriers that are rapidly encountered. So the magnitude of diffusion changes with direction in space, and that phenomenon is called anisotropy. Now, in around 1995, about the time I started my PhD, a new diffusion imaging technique called diffusion tensor imaging was invented that allowed us to measure diffusion anisotropy, and it produces two types of image. The first is this mean diffusivity map, which is a so-called directionally average measure of diffusion. It just represents how much tissue structure there is present. And then this anisotropy map, where the bright areas here are areas in the brain where the structure is very coherent and all of those pathways are pointing in the same direction, so we get a coherent structure. Now, when I was doing my PhD, I was working in the MS research group at the Institute of Neurology, and I worked very closely with this chap here, David Waring, who's a consultant neurologist with his own research team now at University College Hospital. And we produced at the time what was the first DTI maps in multiple sclerosis, which is a disorder that involves a degeneration of the myelin sheath, which is wrapped around the axons and facilitates nerve conduction. And you can see here that on this mean diffusivity map, we've got an increase in diffusion because there are fewer barriers to the diffusion process there. And this lesion is sitting in the white matter tracts of the brain, so we've also got a reduction in fractional anisotropy. So these methods provide us with a quantitative measure of how severe the structural damage to the brain is when, for example, lesions occur, such as those in multiple sclerosis. Now, the most important paper that I published during my PhD was this one, where I was able to obtain the first quantitative diffusion maps of the human spinal cord in a living subject. So you can see two images here with diffusion measured roughly along the cervical cord here and perpendicular to the cervical cord in this image here. The thing to notice is that this one is brighter than this one, which tells us that diffusion is greater when measured along the cervical cord than in directions perpendicular to the cervical cord. So this is, again, the phenomenon of anisotropy, because the cord consists of these long, thin structures. Now, these images were quite difficult and challenging to obtain, partly because the spinal cord is only about 12 millimeters in diameter, so it's very small compared to the size of the brain. It's also moving up and down during the normal cardiac cycle, so we've got motion issues, and also it's surrounded by this bony structure, the spinal column, which tends to distort the uniformity of the magnetic field. And a distortion of the magnetic field will lead to a geometric distortion in the images. So eventually I completed my PhD. Here I am celebrating at the Queen's Larder, just round the corner on Queen Square, and with my wife Catherine, who obtained her PhD at the same time, on the portico of the main UCL building. So following that, I was fortunate enough to be offered a postdoc position in Orsay, just south of Paris in France, by this man, who's widely regarded as the godfather of diffusion MRI. And I think, he was quite tickled to have a young Englishman working in his laboratory. It was an amazing experience to live and work in Paris. The food, as you might imagine, was amazing, including the food that we had in our staff canteen. We used to get foie gras at Christmas, which was a particular treat. There were some interesting cultural differences to observe. For example, all MRI scanning of human subjects was suspended for the month of August. And I also remember that they were quite reluctant in sending their papers for publications. They kept tinkering with their methods and techniques, I think, so that I could perfect my masterpiece. So this is an early paper where we perform what's called tractography, i.e. we're going to map connections between one brain area and another by following a pathway through the white matter of the brain, as we shall see. So let me explain. So from the imaging data from diffusion tensor imaging, we get a vector field. So in each pixel of the brain image, we've got a little vector telling us the direction along which diffusion is greatest. This represents pretty much the direction of the white matter tracks in that location. And tractography is where we attempt to draw lines of connectivity through this field, as you can see here, and reconstruct the white matter tracks of the brain. So this was a very early attempt to do that. What you can see here is an image showing you an attempt to track the motor pathways of the brain, pathways connecting the primary motor area up here, the so-called seed region, down to this target region in the brainstem. And you can see we've reconstructed this pathway. This was called a spaghetti plate algorithm because it's a type of algorithm called an energy minimization algorithm. If you take spaghetti and you plunge it into hot water, the hotter the water, the more you can get the spaghetti to bend. So what we're trying to do here is minimize the energy and obtain the smoothest possible path through this field. And this actually has got one of my favourite equations, E spaghetti equals this integral here. Okay, so the most important paper actually published during my time in France was this one on water diffusion, compartmentation and anisotropy in the human brain. And what this really did was to recognise that the Einstein equation doesn't actually fit the data very well. So what you can see here again is a plot of the log of the signal versus the B factor, and you can see that it's no longer looks anything like a straight line. So what I was trying to do here was to recognise that in fact water diffusion in tissue is taking place both inside cells and outside cells. So it's recognising that there is an intracellular water diffusion and an extracellular water diffusion. And you can fit, I fitted at the time this model, which is a slightly more complex model with two exponentials now, one representing a slow diffusion compartment and one representing a fast diffusion compartment. I then moved to the Lucas MR Center in Stanford in California to continue this work and extended it to measurement of the fast and slow diffusion tensors. And so there's the equation now. These become diffusion tensors up here. And I obtained this image, which was quite interesting. This is a mean diffusivity image of the slow diffusion fraction. Now, what you can see here, in contrast to the conventional mean diffusivity map, is the emergence of contrast between white matter and grey matter. So all this stuff here that looks quite dark is in the white matter. So that's telling us that slow diffusion is slower in white matter than it is in grey matter. And this is due to the presence, presumably, of myelin in the white matter, which doesn't exist in the grey matter. Now, these maps do look a little bit ropey. You can see there's a little bit of noise there. Well, one has to remember they're actually 14 years old, and MR technology has moved on an awful lot since then. Now, I started my own research group and obtained my first research grants when I moved from Stanford to St George's Hospital Medical School, as it was then called, in dear old Tooting. So it's a little bit of a culture shock going from Paris to California to Tooting. No disrespect to my colleagues there. I can see Tom there laughing at me. However, Tom was my first recruit, a talented young scientist, and together we produced some very contemporary work in tractography, a field which was still very new at the time, but was blossoming. So I programmed the MRI scanner, and Tom coded up this really nice tractography algorithm. So what you can see here are streamlines of the sort that I was showing you before, and each of these streamlines represents the white matter pathways in that location in the brain. They're coloured according to their orientation, so green, anterior, posterior, blue, superior, inferior, and red are pathways running from right to left. Now, this image won an art competition, and it's funny that it keeps cropping up in different places. In fact, when I searched the internet recently, from Fine Art America, nervous system iPhone cases. How about Brain Pathways by Tom Berg and Chris Clark, only $49.95. So, the good old British pub. We started to have quite a lot of interesting and fruitful scientific discussions in a good old boozer. And quite a few of our favourites at the time was the King's Head, Hoochie Mama's, which was a little bar on Tooting High Street, and the Exhibit, which was a trendy new bar in Balham. Now, let's just look at this a little bit more closely. Here I'm going to plot creativity, scientific creativity, against number of beers consumed. Now, the first data point there you notice is actually not on zero. You've got a small effect there of just the mere anticipation of going to the pub raises one scientific creativity. But we found that creativity tends to peak around 2 or 3 pints. And it's really important, I should say, that at this point you must write down. You really do need to write this down, because after that things go pretty horribly wrong. So we actually ran this experiment quite a few times, highly reproducible findings. I was then fortunate to link up with Tony Bell, who I'm delighted is here this afternoon, who is the Professor of Neurosurgery at St George's. And I basically published this paper, which was really amongst the first to look at tractography as a tool for neurosurgical planning. So the idea here is that the surgeon wants to remove a portion of the brain or a particular lesion or a tumour, and in doing so they want to avoid damage to eloquent white matter pathways of the brain. Now you can see immediately from this that with tractography we're now able to visualise those pathways and provide that information to the surgeon, helping them to avoid post-surgical deficits. So hopefully this video illustrates what I mean. It shows a reconstruction of the corticospinal tracts. Tom will probably remember this, it's quite an old video, he's laughing there. And what you can see is this tumour shown in red and this pathway wrapping around that large mass. So we can say that those pathways are intact, but they're in close proximity to that tumour. However, in this second example, this is a patient with an astrocytoma, shown in red again. You can see that pathway sitting right in the trajectory of the motor pathways. In this case, it left the patient with a motor deficit, a hemiparesis. So one day, Tom and I took a walk into the bowels of the medical school, and amongst the murk and formaldehyde, we uncovered Nigel Laws, who I'm delighted is here this afternoon. Nigel was one of a dying breed of rare scientists, neuroanatomists. And I remember when we first showed him tractography, he was really taken aback and said something like, "Wow, I've been waiting my whole life for something like this." And he certainly really did make the most of tractography, as we'll see. And you could argue that tractography has really brought about a renaissance in neuroanatomy ever since. So this is really one of my favourite papers, where this was work led by Nigel, comparing the results that we get from tractography with blunt dissection of tracts from post-mortem brains. So what he did was to painstakingly label the brain surface, as you can see here, all of these different labels, and perform whole brain tractography, looking at clusters of connections, streamlines between these different brain areas. And when you do that and you project those different clusters onto the surface of the white and grey matter, you get these really beautiful images, as you can see here. And those of you in the audience that are clued up on neuroscience will recognise that these look an awful like Brodmann maps. And Brodmann maps have been around for over 100 years. So these are maps where each of these little regions can be segregated according to the appearance of the cells that you see under the microscope taken from that area of the brain. And those regions correspond more or less to some of the different functional specializations of the brain. Now, Nigel undertook a very exhaustive and rigorous comparison of tracts isolated from post-mortem specimens with tracts isolated virtually with tractography. As I said, here are some figures from that paper, and found a very close correspondence between the two. And this figure probably illustrates it best. This is a tract called the inferior fronto-occipital fasciculus. Here's the blunt dissection, and you can see arrowed all of these different morphological features that are reproduced very faithfully in the tractography. So this study provided a lot of, if you like, validatory evidence for the value of tractography, giving us confidence that the results that we get from tractography are actually meaningful. Now, we know that some functions in the brain are specialised and lateralised, such as language, for example. We tend to use more of our left hemisphere for language than we do our right hemisphere. And this raises the question as to whether the white matter tracts that underpin some of these functions are also lateralised in the brain. So this was work that was led by Tom Barrick, And you can see here that he's reconstructed pathways connecting the temporal and the parietal lobes. And this particular group of pathways is bigger in the right hemisphere than it is in the left. This is the so-called superior parietal lobule. And in common with other studies around that time, we also found that the arcuate vesiculus, which is this curved structure here in the right hemisphere, and here it is in the left, it's heavily involved in language, as we heard in the previous talk from Torsten Bordeweg, and it's larger, as one might expect, in the left hemisphere than it is in the right. But this was confirmed using tractography. Now, in 2006, I returned to UCL, this time to the Institute of Child Health, whose mission statement is to improve the health and well-being of children and the adults they will become. Now, the first thing I realised, having up to that point in my career only worked with adults, is that working with children is just, well, it's a lot harder. Because, I mean, I don't have to deal with this personally, but a lot of my clinical colleagues next door at Great Ormond Street Hospital will tell you, you have to deal with very concerned and anxious parents. Children are quite nervous about the MRI scanner. Little ones have to have a GA to be able to go in the scanner. But from a completely scientific point of view, you're now dealing with brains that are changing markedly in size and microstructure, going from the neonatal period through childhood and onto adolescence and young adulthood. So a feature of our work, as I probably mentioned a minute ago, is our close relationship with Great Ormond Street Hospital. Here's our 3D MRI scanner, which was recently installed there a few months ago. We're very excited about that. It's my pleasure to lead the developmental imaging and biophysics section, which is a multidisciplinary team consisting of people working on image methodology, imaging methods, through to applications of those methods in a diverse and wide range of conditions, such as epilepsy, neuro-oncology, and sickle cell disease, amongst others. Now, in the intervening years, the quality and power of diffusion MRI has improved markedly. These are some of the most beautiful images. My colleague Donald Tournier produced these. I think he's somewhere there in the audience. He's probably smiling happily to himself right now. These are really lovely. They have they show how we've managed to solve the problem of crossing fibres, which you can see up here. These pathways are actually crossing over each other, which can't be resolved using diffusion tensor imaging. So that was a coronal view. This is an axial view, a sagittal view. And I think it's true to say that tractography has captured many people's imagination. And as you can see here, the band Muse have put it on the front cover of their album. This is a particularly delightful video, I think, courtesy of Matt Rowe and Danny Alexander from Computer Science at UCL, which shows tracking of the cingulum bundle, which is a little pathway that sits just above the corpus callosum, which joins the two hemispheres of the brain together. And you can consider that in this way, science is almost becoming like art. And this leads me to a meeting I had with an artist who had seen these images and asked me to be involved in a project called Fisher. And that artist was Louise Ann Wilson, who I'm delighted is here this afternoon. She's a UK-based scenographer making site-specific performances. There's Louise. And the project explored ideas about mapping the flow of water both in the brain and in the landscape, represented through music and dance, including my descriptions. I wasn't dancing, don't worry. Including my descriptions of mapping water diffusion in the brain using MRI. So I was both a performer, contributor, and a member of the audience at the same time. And it was a truly unique experience and one that I'm never going to forget. Now, we know, of course, that boys and girls are different. Here are my three, Hannah, Freddie and Isabel. But what about their brains? Are there differences in the development of brain microstructure between boys and girls? Hello, Hannah. So this was a collaboration with Faraday Varga Kardem here at the Institute of Child Health. Now, what I've got are two graphs. Here we've got anisotropy plotted against age. Here we've got the mean diffusivity plotted against age. So remember, anisotropy represents the coherence of the underlying white matter pathways, when they're all lined up together, we get a high value of FA. MD represents the overall amount of tissue structure present. Now you can see that we've got the boys here in blue and the girls here in red. And what you notice, particularly when you look around 8 to 9 years of age, is that the boys have got a less anisotropic structure than the girls, and then it tends to converge around 10 to 11 years of age. Whereas for mean diffusivity, the overall amount of structure present, it's higher, indicating less microstructure for the boys than for the girls around this age. So there are real differences in these developmental trajectories between boys and girls, and we think that this is probably related to the later onset of puberty in boys compared to girls. So here is a dissection of the brain again, and you can see what I want you to focus on here are the optic radiations. So this is an important part of the visual system. So here we've got the thalamus, and the optic radiations curve out from that structure and connect into the occipital cortex. This is called Mayer's loop here. And you can see that with tractography, we can do a pretty good job at reproducing those pathways by imaging somebody's brain and reconstructing those pathways using MRI. So one of my former PhD students, who's now working in New York, undertook to measure these anatomical distances in children. And here's an example of that here, now shown in a sagittal view of the brain. Here are the optic radiations in red and yellow, overlaid onto this image, and you can see that he's able to measure this distance between Mayer's loop and the temporal pole. Now, again, this is useful and interesting for the reasons that I was talking about before with regard to neurosurgical planning, because patients with epilepsy that doesn't respond very well to drugs are offered a type of surgery that involves removal of part of the temporal lobe with a view to trying to render the patient seizure-free. And so if we can provide this information to the surgeon, you can see that we can avoid the surgeon damaging this pathway, which can lead to a visual field defect. And then on the same theme, here are a series of slices showing the optic radiations on these T1 weighted MRIs. And you can see the close proximity of that pathway to this large occipito-temporal lesion there that the surgeon's going to operate on. It's been supposed that most of the connections from thalamus to the visual cortex connect to the primary visual cortex, so-called V1. And in this work that was done by Ivan Alvarez, another of my PhD students who's recently moved to Oxford, he combined tractography with functional MRI to show that there are significant connections to higher visual areas V2 and V3. So what you can see here is a reconstruction of the optic radiations. Connections to V1 are shown there in red, but there are significant connections in yellow to V2 and green to V3. So coming back to the thalamus again, this is where we've zoomed in on this structure, the thalamus, which has many connections to different areas of the cortex. And what we've performed here is what's called a parcellation. So each of these colours that you can see here represents connections from that part of the thalamus to these different cortical targets. And they're coloured according to the combinations of cortical areas that they connect to. So the purple areas here will have connections to multiple cortical regions, whereas the red areas here only connect to relatively fewer cortical regions. And what you can see here on the top row, these are healthy controls, and these two lines here are patients with a genetic neurodegenerative disease called Huntington's disease. And what you can see is that before the onset of symptoms, there are very subtle changes to these patterns of connectivity, and they become much more pronounced when the patients proceed to the clinical manifestation of the disease and suffer from neurological deficits. Now, sickle cell disease is a common genetic disorder that occurs when red blood cells are unable to carry oxygen effectively around the body due to the sickling of their shape, as you can see here. Patients suffer from low blood oxygenation, recurrent pain crises, cognitive problems and clinical strokes in about 10%, and asymptomatic events in about 30% of the sort that you can see here, little lesions like that. Now, this is a collaboration with Fenella Kirkham and one of my PhD students who recently obtained her PhD, Jamie Kawadla, where she is demonstrating here a correlation between what we call the radial diffusivity, diffusion measured perpendicular to the white matter axons of the brain, and the degree of blood oxygenation and hemoglobin. So what this is telling us is that chronic, persistent, low blood oxygenation is leading to a degeneration of the brain structure, and it seems to be prevalent in the corpus callosum, which connects the two hemispheres of the brain together, and we need for many cognitive tasks. Now, there's a large sickle population in Africa. This is a photo from a recent trip I made to the Muhimbili Hospital in Dar es Salaam in Tanzania, where we're training Dr. Mboka here in this very brightly coloured, delightful dress. And she's a young radiologist. Now I am sitting at the console of their MRI scanner. So I just want to just take a minute to consider, what is it actually like to work in research? And the best image I could find that I think represents this is this. So it is a little bit of a roller coaster ride. Certainly, those of you, my academic colleagues in the audience, I'm sure will sympathise with this idea. But you work really hard, eventually get your PhD with much relief and joy, and then you send your paper for publication, and it gets rejected by the journal, and it's a very chastening and disappointing experience. But of course, you dust yourself off. You revise your paper, send it off to another journal until eventually it gets accepted. And the same process happens, of course, for those of us who have been in science for longer and are now what we call principal investigators, obtaining our own research grants. We spend months and months working on a grant, and then most of the time it doesn't get funded. I can see a few people nodding in the audience. It is a real pain sometimes. But that's how it is. I think one observation here is that hopefully these oscillations, these ups and downs, actually start to taper off a little bit as we get older, a little bit more long in the tooth. Although I must say, with the best of them, I am capable of a blaspheme should one of my grants not get funded. And equally, I'm happy to accept a glass of champagne should one of them actually succeed. Now, my last little bugbear that I just want to mention here is this issue of why we still hand over copyright and publications to scientific journals. So we spend a lot of time working on our papers, send it for publication, and eventually it gets accepted, the paper gets published, and we have to pay the journal for the paper to be freely available to everybody. And in addition, we often have to pay the journal, sometimes several thousands of pounds, to reproduce colour figures from the paper. And also we sign over all the copyright of the work, even though it's all our own work. Now, my view on this is that, to a certain extent, I mean, I know that a lot of us here are wedded to journals, and many of them have been around for about 100 years and so on. But do we really need journals and publishers anymore? I think the custodians of the copyright of the science that we do should be this institution, University College London. And all we need to do effectively is to obtain a pool of peer reviewers and a number of academics that are willing to stand as editors, and we should be able to publish our own work online, freely available to all under the banner of this institution. Okay, let's rant over. Let me return now to brain tissue microstructure. So what you can see here are some electron micrographs. These are myelinated axons that are all pointing towards you. These are only about a micron, few microns in diameter. That's the myelin sheath wrapped around the axon that helps nerve conduction, which I mentioned earlier. And here you can see an axon with a really large diameter. Now, an interesting parameter is this so-called G ratio, which is the ratio between the diameter of the axon and the surrounding myelin sheath. If you look on electron micrographs, the G ratio tends to come out at about 0.6, which represents an optimal value with regard to conduction velocity. Now, the G ratio, as we would expect, will change during normal brain development, but it's been suggested that it might underpin differences between boys and girls, and also conditions like, for example, schizophrenia. Now, this is work that was done by Ollie Gould, who did a short project with us, and what we've got are two types of images to begin with: a measurement of the myelin volume fraction, which we can get from a technique called magnetisation transfer imaging, which has been around for years, and then advanced diffusion MRI methods, such as NODI, which was developed by Gary Zhang, who's in the audience here at UCL, which can gives us maps of axon volume fraction. So the contrast in these images is actually inverted. The dark areas have got high myelin volume fraction, and the bright areas have got low myelin volume fraction, and they can be combined to create a so-called G ratio map, which is what you can see here. So I think this is really exciting, a new contrast that we can actually obtain from MRI in living human subjects. And just focusing on this in a little bit more detail, here's a sagittal view, and you can see this darker area here in the splenium of the corpus callosum. That's exactly where we expect to find those giant axons with very large diameter. Okay, spatial problems with spatial navigation. This is a personal story. So we know that the brain is good at telling us where we are and how to navigate the space that we're in. This is the pioneering work that was done by John O'Keefe, who won the Nobel Prize last year. John O'Keefe is here at UCL. And he discovered these cells, called place cells in the hippocampus. These fire when the rat passes through this location in this environment. And he shared the prize with his Norwegian colleagues, who showed that these grid cells in the entorhinal cortex fire when the rat passes through any of these blue regions here, organised in a grid fashion. Now, one assumes that this neural mechanism exists in humans. This is the SECC. It's a large conference centre in Glasgow, where I was about to give a talk at a conference quite a few years ago. I was feeling a little bit nervous, and I was sitting there, and unfortunately I thought to myself, oh, you know, I need a wee. I do actually need a wee. What am I going to do? The clock was ticking down, and I made a snap decision to run to the loo. So I headed for the exit, went through the exit, through some double doors, turned left, turned right, down another load of steps, and so on, until I found myself in the bowels of this huge conference centre, and went to the loo. Oh, that was good. But I was conscious at the time. I was thinking, oh my God, you know, I've got to get back there in a couple of minutes. And then I suddenly realised I didn't know how to get back to the auditorium, which was a crushing blow. And I was feeling really panicky at this stage. And this is the auditorium that I was speaking in. And I started to run around like a headless chicken, and my grid cells and place cells were misfiring all over the place. And until eventually I realised the best thing I can do is head for the exit. So I headed for the exit, and I came out down these steps, and I came out right at the back of the SECC here on the street. And then I had to run all the way around the conference centre to the front entrance here, and smile politely at the people on the door, show them my badge, and then ran down to the front and managed to get there just in time as the chairman said, and the next talk will be given by Dr. Clark. So that's my problems with spatial navigation. This is a map which we all know and love, the tube, which is a map, which is a network and represents connectivity. It doesn't really convey distance very well, but it conveys connectivity. You can see that some parts of the network are very highly connected and have got lots of pathways running through them. We can use a representation like this to encapsulate the connectivity of the brain. So we can do that by identifying the stations through parcellation of these different specialised cortical regions. So we've got computer algorithms that can identify these, and they're labelled with different colours. Then we perform whole brain tractography, which allows us to infer connections between these different nodes, these different cortical nodes, and from that we can create what's called a graph. So it looks a little bit like our tube map. So here we've got different cortical regions and the lines represent the connections between them. So we can interrogate that graph and it tells us about how efficiently information can move around the brain network. Now we can do a similar thing with a technique called resting state fMRI. I haven't got time to explain it now, but essentially it's a method where the signal fluctuations that we observe are related to changes in the oxygenation state of the blood, which changes when neurons fire. So this is a mechanism for measuring the functional connectivity of the brain. And in the same way, by doing parcellation of the cortex and so on, we can build up a graph of the functional connectivity of the brain. And in normal, healthy individuals, the structural connectome, as it's called, and the functional connectome are very, very closely related. They're very closely correlated with one another. But what we observe, which is interesting, is that that correlation breaks down in neurological disease. So this represents an interesting new method that is available to us for the investigation of neurological dysfunction. Okay, I'm nearly at the end, and I'm sure you're all gagging for a drink outside. This is the last bit of science that I'm going to talk about. Duchenne muscular dystrophy is a degeneration, causes a degeneration of muscle due to a mutation in the dystrophin gene. So patients have difficulties in walking, a higher risk of neurobehavioral disorders such as ADHD and autism, and a severely shortened lifespan. Now, a typical way to monitor the progress of this disease is to obtain a muscle biopsy and look at the biopsy under the microscope. Now, of course, that's a very invasive procedure and it can only be done so many times. So you can imagine that we're very interested in trying to get the same kind of information from MRI. And we can use those methods to monitor as many times as we like treatments such as those developed by Francesco Montoni and colleagues here at ICH. So this is our sort of first attempts to do this. It's kind of a new field for us because we've all been working on brains for so long. This is work that's been led by one of my postdocs, Matt Hall, who identified this geometry. So if I just go back, to the muscle structure, it's got this property where we've got a structure within a structure, within a structure, within a structure, this kind of nested effect, which you don't see in brain. And that can be quite nicely represented by that geometry, which Matt identified, which is called a recursive Apollonian gasket. So this is basically a fractal structure. And what we can then do is perform a simulation where we can simulate that structure, and each of these little red dots is what we call a random walker. So that's a water molecule, and we can use that computer algorithm to allow those water molecules to execute a random walk through this structure, and it allows us to work out what different aspects of that geometry we can recover from diffusion MRI measurements in living subjects. So I want to finish now by thanking a few people, and I'm going to start with my parents are laughing, smiling, and oh no, Mum, it's not that bad. This is from their wedding day, 1963. Is that right? Have I got that right? Yeah, it is 1963. And I like this picture partly because my dad looks like a teddy boy. I'd like to thank my wife, Catherine, who's been on an academic journey of her own as a radiotherapy physicist. Thank you for all your love, all your support, listening to my occasional rantings from time to time. And of course, my lovely kids, Hannah, Freddie and Isabel. Research, of course, isn't possible in isolation. It's always a team effort. I'd like to thank all of the current members of the Developmental Imaging and Biophysics section here at the Institute of Child Health, which I'm very proud to lead. Tina Banks, our research radiographer, and also Sally Dowsett. I didn't get a picture of Sally, unfortunately, in time, but she was our long-time administrator in the unit, and she retired last year, so thank you to her too. All of my former PhD and MD students, all of them have stayed in science and medicine, so I can't be that bad a supervisor, hopefully. And finally, I'd like to thank these four people in particular. Gareth Barker and Paul Toffs for teaching and nurturing the young PhD student, Denis Le Bihan for his inspirational approach to science. He's got some pretty wacky ideas, but he's got some very good ones as well. And finally, David Gadian, here from ICH, for all his help and guidance on my path to becoming UCL professor. I'm really proud to have worked with all of these people, including many other collaborators and colleagues that I've not shown here, both from across ICH, UCL, the UK, and worldwide. Most of all, I'm proud to count myself among them as a member of the world scientific community. Thank you.
Well, thanks very much, Chris. A great talk. Thank you indeed. For me, it's been a brilliant afternoon because it acknowledges the contributions of two outstanding colleagues of mine, Torsten earlier and now Chris. So it's really lovely to see how things are moving forward. Chris, what does one say to somebody who's taken over from me as head of unit? Well, you know what happens with regime change. Things happen, especially if there are what some may regard as profound differences between the former and the latter. There are people in this institute who, if they're perceptive enough, will see that there's a picture missing from our labs, offices upstairs. You know what happens to art with regime change and culture. Some of you will know what that picture is. It's the picture of the 1999 Manchester United treble winning team. And it's no longer there. Why is that, you might ask? Well, actually, I removed it myself. It was a 50th birthday present to me from our group. And I felt I had to put it in safekeeping. It's now with Peter at the front desk, who is also a Man United fan. Because, as you know, Chris, he didn't mention it, I don't think, but Chris is an Arsenal supporter. And it's with a very, very heavy heart that I have to say, well done in getting to the semi-finals. Some may know that they beat Manchester United. But anyway, when Chris took over from me as head of unit, to be honest, it was a brilliant move I think it was great for me because it gave me the opportunity to focus on some research that I started up in Nottingham. And it was great that the younger man, Chris, had the vision and everything else that it took to take the group forward for the coming years and possibly decades. He's put together a great new team, a very vibrant team, by my standards, a very useful team. And it's been, for me, really lovely to see. And I think this, you can all see now through just some of the work that Chris has presented in this really fascinating talk. He's done incredibly well in building up the unit for the present and for the future. And he's done really well in bringing in funds, in establishing collaborations locally, nationally, internationally. And we can all look forward to a bright future. So I'd just like to say, Chris, many thanks and well done and many congratulations on everything you've achieved. And let's all join together in thanking you and congratulating you and wishing you all the very best for the future.
Thank you all. all very much for coming. I'm sure you've all had a wonderful afternoon and I really enjoyed it. I've really enjoyed it personally. I think it's been lovely. And there was one thing that Ross was supposed to tell you at the beginning, and I was supposed to tell you, so I'll tell you now, is the lectures were being recorded. So if you didn't notice, that's okay. Anyway, I'd like to now invite you to come out on the balcony and join us for a drink. And once again, thank you all very much.